Hey everyone, my name is Jonathan White, and today I'm going to be showing you 3 different ways to transfer bacteria using the aseptic technique for growing bacterial cultures. So, step 1, you're going to need to put on your PPE, so gown, gloves, and goggles. And then you are going to turn on your micro incinerator, which is the toilet paper right here. Turn it on. It takes about 10 minutes for it to warm up. While this is happening, you want to sterilize your workplace. So I have cleaner here, spray, sterilize alcohol cleaner, killing off all the bacteria so you don't cross-contaminate anything. I'll put that over there. And then you want to make sure that you have your materials ready for this experiment. I have 2 plates, Apgar plates, and then 2 test tubes right here, and I have these guys pre-labeled, so these guys are labeled with label tape, and then my 2 petri dishes are labeled on the bottom. So the bottom of the petri dish is the smaller part right there, and I want to label around the edge of the petri dish, and I want to make sure that I have my name, the date, and the species of bacteria. And then I also need an inoculating loop for this experiment, which I have here. So after the micro incinerator has been warming up for 10 minutes, it's time to sterilize the inoculating loop. So you want to do that by holding it in the micro incinerator for 10 seconds. And when you do this, you want to make sure that you're holding the entire part of the inoculating loop, or the entire wired section of it, in the incinerator. So you hold it in there for 10 seconds, 1, 2, skip a few, 10, and then you let this cool down for a minute. You do this so that it's not too hot when you go to collect the bacteria. If it's too warm, it might kill the bacteria that you're trying to test. Now that it's cooled down, we can start to transfer bacteria. So what I'm gonna do is grab the test tube that I'm gonna transfer from, and then I'm going to grab it in the body of it on the glass, give me a really firm grip on it, and then with my hand with the inoculating loop, I'm going to use my pinky and uncap this test tube, just like this. And now that this is opened, I want to sterilize the lip of this for about 2 to 3 seconds, just so no particles get in there. So I hold this close to the micro incinerator for 2 to 3 seconds, and then once that's done, I can take the inoculating loop and collect a bacteria sample. So I go in there, collect the bacteria sample just like this, and I want to sterilize this one more time. So I'm going to hold it here, sterilize it for 2 to 3 seconds, and then place the lid back on nice and firmly and place upright in my upright container. Now that this is done, we can transfer this bacteria. So I'm going to grab the second container that's been pre-labeled, that I'm going to, or broth basically, or second test tube with a broth in it that I'm going to transfer the bacteria to, making sure that it's the right one. With my finger, my pinky finger, I'm going to uncap it, and then I'm going to sterilize it right here for 2 to 3 seconds. And once that's done, I'm going to transfer my bacteria with the sterilizing tube into it. So I'm going to hold it down just like that, and then shake it back and forth just like this, making sure to get all the bacteria off into it. What I'm going to do is pull this out, hold it close to the incinerator again, because we want to sterilize the top of it, 2 to 3 seconds. And then you're going to place that cap back on, just like that. Then you're going to put that back in there. And then you are going to clean this. The way you do that is by sterilizing it in the micro incinerator. So you're going to hold it in there for about 10 seconds. Okay, now if we wanted to transfer to a petri dish, we would do everything the same. We would sterilize our inoculating tube. We'd grab the sample that we were going to take from, open the cap with our pinky, sterilize the cap, collect our sample, sterilize the lip of the sample tube, replace that back, and then what we're going to do is we are going to take the petri dish that we are transferring this bacteria to, and we're going to open it like a clam. So top is the larger portion, and we're going to open it just like this, and we are going to take that sterilizing loop, and imagine this having agar in there, a red substance along the base. We're going to take the loop, holding it parallel, not too steep, but parallel, just like this, and we are going to gently scrape or touch across the surface of the agar, making sure that you're not going to puncture it, anything like that. And then we are going to close that lid, and we're going to place this upside down in the incubator chamber. And we place it upside down just in case any water droplets form. They will not contaminate the bacteria. So now that that's placed in the incubator, we are going to sterilize our loop by placing it back in the incinerator for 10 seconds. In this final video, I'm going to be transferring bacteria from one petri dish to another petri dish. So making sure that I have all my PPE on, gown, gloves, and goggles. I'm going to turn on the micro incinerator here. It needs to warm up for about 10 minutes. Then I'm going to clean my workstation. So spray, 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 wipe, wipe, wipe. Making sure that my petri dishes are labeled on the bottom correctly. Name, date, organism that's inside of them. What I'm going to do first is take my inoculating loop, and I am going to sterilize this after this is warmed up for 10 minutes. So I'm going to hold the inoculating loop in the micro incinerator for 10 seconds. And once that's done, I can pull it out, and I'm going to let it rest for a minute. The reason you want to let it rest for a minute, it's so that it's not too hot that it kills the bacteria. And then I am going to take one petri dish with the bacteria swallowed inside of it. I'm going to open it halfway like a clam, and with my now cooled inoculating loop, I am going to slide that through here and gently take a sample of bacteria. You don't need a lot of solid bacteria, just a smidgen, maybe half the inoculating loop there. And then I'm going to place that down. And then my second petri dish, I am going to open partway, and then take the inoculating loop, slide it in here, and gently slide across the surface, making sure that I'm not at an angle where I can dig into it, but gently touching, just like that. And then I'm going to close this lid, just like that, and I'm going to place it upside down in the incubation chamber. And when that's done, I am going to sanitize my inoculating loop by placing it back in the micro incinerator for about 10 seconds, holding it there. Now that that's sanitized and both of my petri dishes are where they need to be, I am going to clean my workstation one more time. So spray, 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 spray. Clean, 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 just like that. Turn off my micro incinerator, put away my PPE, throw away my gloves, and put all the stuff back. Thanks for watching.